Namaste to all students. Khushbu Vidya, myself Jivin, Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about plant tissue culture and their techniques. And this session is related to Plant Biotechnology course of program MSc BT second sem. So, plant tissue culture is a technique of growing plant cells, tissue, organs, seeds, or other plant parts in a sterile environment on a nutrient medium. If you are talking about the history related to the plant biotechnology, so in 1902, the idea of totipotency of plant cell was given by Heberlen. In 1937, White first time established successful root culture of tomato. In 1941, whenever Beek used coconut milk for the growth and development of young Dhatura embryos. In 1957, Skoog and Miller demonstrated the role of auxin and cytokinin, which are plant growth hormones, on root and shoot formation in tobacco tissue. In 1962, Murashi and Skoog, these two scientists introduced the media for tobacco culture, and now it is very famous media known as MS media. In 1987, isolation of Bt gene from bacterium Bacillus thuringiensis has been done. So these are the highlights which are related to plant biotechnology. And if you are talking about the fundamental principle behind plant tissue culture, so plant tissue culture technique basically depends on two concepts, two major key points. First one is totipotency and second one is plasticity. Totipotency is the ability of plant cell to regenerate into a whole plant. Means if any organ or any small part of plant which is having a capability to just cell division and to regenerate and to prepare complete whole plant, so this ability is known as totipotency. On the other hand, plasticity is the ability of plant to alter their metabolism, growth, and development to best suit their environment. There are some terminologies which you have to know before starting plant tissue culture technique or before starting experiments of plant tissue culture. First terminology is explant. What is explant? So plant tissue culture are generally initiated from multicellular tissue fragments called explants obtained from living plants. Explants may originate from a wide range of a plant tissue such as leaf, stem, root, petiole, hypocotyle, cotyledon, embryo, or meristeme. You have to choose young plant part for the regeneration process. How you will select the explant? So explant is selected. It is either haploid or diploid explant. The plant growth can be achieved in two ways, like shoot directly by appropriate media, or by second way is by somatic embryogenesis. So whenever you are choosing your explant, so just make sure that region or part of plant should be new or newly emerging portion should be selected for explant. And after the selection of explant, you have to go for the sterilization process. So sterilization method used in plant tissue culture laboratory, all the material like vessels, instruments, medium, plant material, etc. used in culture work must be free from microbes. Means during the sterilization process, first of all, you have to clean explant with a tap water, in running tap water actually, for at least half an hour. And after cleaning all the dust and other waste material from your explant, you have to go for sterilization process by sodium hypochlorite 2020 or Triton X100 you can also use. So by washing with these chemicals, you have to wash again your explants with autoclaved sterile distilled water. by 4 to 5 times. After cleaning with these chemicals, you have to put your sterile explant into laminar airflow, and then you have to go for final sterilization process with mercury chloride. After cleaning with mercury chloride, you have to wash your explants 5 to 6 times with autoclaved distilled water. This final sterilization process must be done in laminar airflow to avoid any contamination. Sterilization process is achieved by one of the following approaches. You can give dry heat treatment, flame sterilization, autoclaving, and filter sterilization, wiping with 70% ethanol, and surface sterilization. These all are sterilization techniques are related to your glasswares and other equipments which you will use for the plant tissue culture technique. Now come to the culture media which will be used in plant tissue culture technique. So explants are, after sterilization process, then usually placed on the surface of solid culture medium, but are sometimes placed directly into a liquid medium when cell suspension culture are desired. Culture media are generally composed of inorganic salt plus a few organic nutrients, vitamins, and plant growth hormones. Before inoculation of explant into culture media, you have to sterilize that culture media also by autoclaving them, and all the equipments like measuring cylinder, beaker, forceps, scalpel, whatever you are using for plant tissue culture technique, you have to sterilize them by autoclaving. As culture grow, pieces are typically sliced off and transferred to a new medium, means subculture. For example, you have inoculate your explant into culture media. After 15 to 20 days incubation period into plant growth chamber, it will be converted if it is going to convert into callus. So after completion of incubation period, you will get complete growing callus, and then you can sliced off some pieces of that culture, and then you can transfer these pieces into new media. So this process means you are just subdividing culture callus, and then you are shifting them into new media. So this process is known as subculturing. So subculturing is used to allow for growth or to alter the morphology of the culture. There are some types of the media which are used for plant tissue culture technique. So first of all, white medium was introduced. So it was one of the earliest plant tissue culture media. Next one is MS medium. It was formulated by Murashi and School, and it is most widely used for many types of culture medium. So it is most successful media for plant tissue culture technique. Next one is the B5 medium. So it was developed by the Gamberg for cell suspension culture and callus culture, and at present it is modified form used for protoplast culture. Next one is the N6 medium. It was formulated by the Chu and used for cereal anther culture. Next medium was niche medium. It was developed by niche and used for anther culture. So these all are types of media in which MS medium is very much famous because it is very much successful, which gave most of the results for in vitro propagation. And if you are talking about the steps which will be followed in plant tissue culture technique, so first of all you have to select the plant, then go for the isolation of explant, and then sterilization of explant by using sterilization chemicals, and then inoculation of the explant into culture medium. But before inoculation of the explant, you have to make sure that your glasswares are sterilized with autoclaving, and after sterilization of your glasswares, you have to prepare media, culture media, and then again go for autoclaving process, means for sterilization process. So after completion of all the sterilization process of glasswares, your culture media and other accessories, you have to go for inoculation of eggs plant. Then incubation period at 25-degree temperature with 16 hours day period and 8-hour night period. This is photo period you have to adjust. And after the incubation period of 15 to 20 days, you will see that there is initiation of callus, means you will get induction of callus. After getting fully grown callus, you can go for subculturing process, means you can slice off that culture into some pieces, and then you can shift or you can transfer these pieces into new culture media. And then you can, when you multiply these callus, you can change the media. You can shift those callus into media which are having hormones for shoot induction. Then shoot will be induced from these callus culture, and after getting fully grown and with more branching of shoots, you can change the media, you can prepare new media for root culture, and then you can just simply cut the branches and then single branch you can transfer into root culture media. This all process means subculturing and change of media for regeneration process, means for first of all for shoot regeneration and for further for root regeneration. This all process will be done in laminar airflow. And after getting complete plantlets with fully grown shoot, with fully grown root, you can go for the hardening process, means you have to shift that complete grown plantlet into coco pit mixture, and this shifting process is known as hardening. And after successful of hardening process, you can transfer the plantlets to greenhouse or to the open field. So these all are steps which basically followed during the plant tissue culture technique. And if I'm talking about the types of PTC, so there are various type of PTC culture which are based on explant used in that particular culture, means seed culture, which use seed for the culturing process, meristem culture, protoplast culture, organ culture, pollen culture, callus culture, cell culture, and embryo culture. There are some factors which affect tissue culture efficiency. Plant regeneration from tissue culture varies with the following parameters. It can depend on plant species, means shrubs and herbs, and especially herbs will give more results, more better results, give better results in comparison to tree. Tree give late results for the induction of callus, induction of shoot, or induction of root. It will take time. But in case of herbs and shrubs, they give easily results in plant tissue culture technique. Genotype within the species also affect the plant tissue culture efficiency. Source of culture tissues means in some plants, like for example, in one plant, shoot can give the better result in comparison to leaf section or root section. So source of culture tissue also can affect the tissue culture efficiency. Age and health of the donor plant means if there is a healthy plant, so it will give a better result. If there is an older plant and health is also not good, so it will delay the results of plant tissue culture. And nutrient medium and other factor also affects, means what is the combination of plant growth hormones, and what is the concentration of nutrient medium. So all the pH also affect the plant tissue culture efficiency. If we are talking about the plant tissue culture application, so the commercial production of plants used as potting, landscape, and florist subjects. It is used to conserve rare or endangered plant species, and especially in those species, plant species which seeds are not viable. And it is also used to screen cells rather than plants for advantageous characters like herbicide resistance or tolerance. It can also use for the large scale growth of plant cells in liquid medium in bioreactor for production of valuable compounds like plant derived secondary metabolites and recombinant proteins used as biopharmaceuticals. It is also used to cross distantly related plant species by protoplast fusion and regeneration of novel hybrid. It is also used to produce clean plant material from stock infected by virus or other pathogens. Next application is it used for the production of identical sterile hybrid species. So you can obtain these species by plant tissue culture technique. So this is the sum up of this session, and this session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University Jaipur. And if you have any query, then mention in the comment box. I will resolve it. Thank you.